Dear student, welcome to the class of basic genetics. My name is Richa and we are continuing discussing the unit 1 elements of heredity and variation. And in continuation, we are going to talk about different ways through which we analyze and understand the Mendel's data. So what were the method that he used may not have the same name as we have today, and how we apply the rules or the laws that we learned from Mendel's data to the other traits that inherit in a Mendelian way. So in the last class we discussed about the probability, we did discuss about product rule, sum rule, and how to solve these genetic problems using binomial and factorial method. So in this class we are going to talk briefly about one more method that help us make a different genetic combination, which is fog line method. We already discussed about Punnett square. I'll just touch upon that briefly, and then our focus will be the statistical method to analyze the Mendelian data. Last time we covered the probability, and today's class I will be discussing about the chi-square test. So Punnett square, as we discussed in lecture 2, it was discovered by R.C. Punnett in 1905. It is a simple grid line method. You can see here we make different grids here, and how we solve this grid is by writing the genotypes of male gamete at one axis and female gamete at another axis, and then we make combination. The details have already discussed in the previous class, and this method is really good to understand when you are very new, but when the things get more complicated, like when the crosses are trihybrid, then it is very difficult to handle these kind of grids. So in monohybrid and dihybrid you would have seen it, and I personally feel that even dihybrid it is taking a lot of time to make Punnett square, but it is really a wonderful method to demonstrate how the traits go together, how they segregate, what are the expected possibilities of them coming together. So without taking much time, I'll first focus on the fog line method. So what is the thing is that how we learn how many combinations when we have dihybrid or trihybrid crosses, more than one traits are there, then how many different type of combination we are going to get, and what will be their probability, right? So as we were discussing in the probability class, it's an important parameter to know if a disorder is recessive, what is the chances of getting that disorder in the offspring and then or getting normal offsprings. So the same way this fog line method help us understand getting how many different combinations possibly we can get, and then what are the probability of getting them. So this method, if I tell you in one line what exactly it does, it actually breaks any dihybrid, trihybrid cross into a monohybrid cross and then solve it accordingly, right? So let's look at the approach step by step. This is just a reminder that when you have a heterozygous parent, you're going to get 3 tall plants and 1 dwarf plant. So 3:1 is going to be the phenotypic ratio, and in terms of probability you will have 3/4 chances of getting tall plant and 1/4 chance of getting dwarf plant. So let's break this dihybrid cross that Mendel has shown, where the seeds were round and yellow, right? So again it is a heterozygous cross. So we have this capital R with a small r and capital Y with a small y, and we are doing a self cross, right? So if I split, as I said, split a dihybrid cross to a monohybrid cross, which means keep the shape and color, which means round and color yellow or green separate, right? You will see it individually. So when you see the shape, so shape is round, and you remember that it is when both recessive come together, it's wrinkle. So when heterozygous, we are crossing heterozygous, then we get 3/4. See, this one thing you have to just memorize, and whenever you are looking at a cross, you see what exactly they are crossing with. If the cross is a cross between two heterozygous, then you already know the ratio is going to be 3/4, 1/4. 4. And if it is a test cross, then you already know that everything is going to be distributed in 1:1:1 ratio. You don't need to really every time write it if you just understand the concept. So when you have heterozygous round, then there are 3/4 chances of getting round seed phenotypically and 1/4 chances of getting wrinkled seed. So I hope you have paid attention to this line. So this line says that this after the R, this line could be R homozygous, RR, or it could be R with small r. So they both make the round seed, right? So that is why we are not really writing if it is RR or small r, capital R, small r, because we really don't know the genotype. That is why the way to write it is R with a line, and so that is round. Same way you can do with color. So with color you have yellow heterozygous cross. In heterozygous cross, 3/4 chances to get yellow and 1/4 chances to get green. Now how this thing is going to solve in terms of the probability, as we were seeing in case of Punnett square. So what we do is we combine these individual frequencies that we get here, the numbers that we get here, with the associated probability, and with the branch method we are going to identify the final phenotypic ratio. So see, 3/4 round and 1/4 wrinkled have equal possibility to go with yellow or green, right? So this 3/4 round have 3/4 probability to be with yellow and 1/4 probability of being going with green, but there are equal chances of going with either yellow or with green, right? That is what I meant. So same way, if you have 1/4 wrinkled, there are 3 more and then you take it with 3/4, this wrinkle could go with yellow and it could go with green, and to be with yellow there are 3/4 chances, and to be with green there are 1/4 chances. So how we are going to solve it? So when we solve it, what we do? We simply multiply the probabilities that we have. So we have 3/4 probability of being round and 3/4 probability of being yellow, and the multiplication of it is 9/16. So round and yellow, getting the round and yellow seed is 9 out of 16. Same way, getting round with green seed, the probability is 3/4 into 1/4, 3/16. And similarly we can solve it for wrinkled, and then you can see the ratio is exactly the ratio that Mendel has, 9:3:3:1. So this is the another method to solve these kind of problems, where we use branch method and we convert every dihybrid cross into a monohybrid cross. Okay, so this way we have solved this problem, and we are getting the same ratios as Mendel got, and we learned in our previous class with the Punnett square 9:3:3:1. So let's solve a problem of the trihybrid cross. So what characters we have for this trihybrid cross? We have round seed and yellow color seed, and we have purple flower. So we have taken 3 characters at a time, and we are doing this heterozygous cross here. And in heterozygous cross, to understand the outcome in the progeny, what we are doing, we are again splitting it into a monohybrid cross. We have seed shape, we have seed color, and we have flower color. So these things, again we know that they are heterozygous crosses, so there are 3/4 chances to get round seed, and there is 1/4 chance to get wrinkled seed, and same is true for seed color as well as for the color of the flower. So this is the part we have already seen in case of the dihybrid cross, where there were the 3/4 round seed could go with yellow seed, and they could also go with the green seed. And when we multiply these probabilities, then we get round and yellow or round and green. Now we have additional factor, which is color of flower. So 3/4 is the probability of getting the purple flower, the same as written here, and 1/4 probability of getting white flower. So this yellow right? Round and yellow combination again can go with purple or white, right? So there are 3 by 4 purple flower and 1 by 4 white flower. Same way this green seed can go with the purple flower or the white flower with their frequencies written. The same way we can solve it for the wrinkled combination with wrinkled yellow and green and purple white. So how we get the final frequencies? We just simply multiply. We multiply 3 by 4, 3 by 4 and 3 by 4. We get 27 out of 65. We multiply 3 by 4 with 3 by 4 and we multiply with 1 by 4. So 3 by 4, 3 by 4, 1 by 4, 9 by 64. Similarly for green, 3 by 4 for round, 1 by 4 for green and 3 by 4 for purple, and what is going to change is these two remain same and 1 by 4 for white, and that is how you can solve it for all the combination, and then you will learn what are the phenotypic probabilities of getting individual combination. So advantages of probability and branch method. Advantages of branch line method is that it helps keep track of all possible genotypes, as I told you before, and these are all the phenotypes that may appear in the progeny, right? And applying the probability, so it is what additional it is doing, it is applying the rules of probability to the branch method, and then it helps us understand the number of progeny that are going to get that particular phenotype, and so this is giving little more detail compared to the Punnett square. So now let's discuss an example to understand the fork line method with the probability methods. So we have the example where we are considering a cross where there are two parents and we want to specifically calculate the probability of one genotype in an offspring. So what is the question? Consider that a cross where heterozygous A, heterozygous B, homozygous recessive C, heterozygous T, heterozygous E in one parent is crossed with heterozygous A's, heterozygous B's, heterozygous C, homozygous recessive D and heterozygous E. And what they are asking us to calculate a probability for getting all recessive traits together in one of the progeny. So I would just want to remind you once again that if you remember the ratios of heterozygous crosses and test crosses, genotypic ratios as well as phenotypic ratios, your life will be very simple. In a heterozygous cross, you remember that there are 1 by 4 and 1 by 4 chances of getting homozygous traits, homozygous dominant trait and homozygous recessive trait. The probability is 1 by 4, 1 by 4, and getting heterozygous the probability is 2 out of 4, right. So 1 is to 2 is to 1, and for test cross the ratios are always 1 is to 1. So you see in all these combination, these heterozygous AA is crossed with heterozygous AA. If you look at B, the heterozygous BB is crossed with heterozygous BB. Same is true for EE. And so the outcome for A, B and E is going to be same, right. So they want us to look at the probability of AA. So what is the probability of getting AA or getting BB is exactly the same, 1 by 4, 1 by 4 for BB as well as EE. And what is the probability of getting this small c together or small d together is the test cross ratio, 1 is to 1. Right, and mean 1 by 2. So we solve this, 2 by 4 is equal to 1 by 2. And so what is true for the CC, you see homozygous CC is crossed with heterozygous CC and heterozygous DD is crossed with homozygous DD. When we get these probability, they are going to be 1 by 4 for AA, 1 by 4 for BB, 1 by 4 for EE, and for CC and DD they are 1 by 2. And because they are the AND events, what we are doing is we are multiplying them. So that is how we solve these kind of problems. So now let's move on to the next part of our talk, where we are going to discuss more complicated data analysis, where we want to compare the ratios that we are getting, if they are the real ones or we are getting something new, if our results are same as expected or we are getting some deviations from the expected results. So remember, in this category of the data analysis, what we have two classes of data, right? So far you are understanding in what probability we are going to get a particular type of phenotype or data. Now we are trying to understand if our observed data is same as expected data or there are differences, right? So now we have two categories, observed data and expected data, and we are trying to understand their ratios. So in many biological experiment we compare the observed ratios with expected ratios, because then only we know that if something, that what I am getting is something which is expected or it is deviating from the expected, and which indicate that there is something happening to the data. So expected ratios, in our case these ratios are based on Mendelian principle, because we are trying to test what? Mendel's hypothesis, and observed ratio are our own data. So observed versus expected may be the same or they may also differ. So let's look at one very neat example of the coin toss that we have taken in the previous class. So if you toss a coin, you know that you have a 50-50% chances of getting either head or tail. But if you toss a coin just for 10 times, do you expect that 5 of the times you are going to get head and 5 of the times you are going to get tail? Probably not. And I would encourage you to try this experiment yourself and see if you do toss for 10 times what exactly you get. So you will see a lot of variability in your data, and if you increase the attempts, if you toss the coin for 100 times, 1000 times, then you will start seeing that this probability is reaching to 50-50 ratio. So when we increase the data, remember the Mendel's strength was the bulk of data. 8 years he spent in collecting so much of data, and if he didn't have that much of data, probably he would not be able to crack these kind of complicated problem. So we need to have significant amount of data to conclude if we have something new or our data is on the expected lines, right? So observed versus expected ratios, if they are significant or not, we can test, right? How we can test? As I mentioned before, we are going to use chi-square test, which is also known as goodness of fit test, to understand our data. The chi-square test assess how well the observed value fit with expected value, right? And what we compare the observed with the expected. So observed is written as capital O and expected with capital E. You can see that chi-square, which is a symbol here, when you sum up all the events of observed minus expected whole square divided by expected, you will get the chi-square value. So this chi-square value is going to indicate the likelihood and the chance alone that could produce a deviation from the expected line. So as I was saying, that you are trying to understand whatever data you are going to get, if it is significantly different from the expected or it is exactly the same, right? So when we analyze such kind of data, what we do, we make hypothesis. And remember, what is the null hypothesis? The null hypothesis is an assumption where we say that there is no difference in expected and observed ratio, right? So if at all I am getting some variation, it might be just by chance, right? So this is one important thing that you have to remember, and we are using these methods, we are only testing the data. How I have collected the data, if I have made any mistakes in collecting the data, maybe my parameters are varying, it's not going to test that. That is something the scientist or the experimentalists need to know, how they have collected the data. These kind of analysis help you make sense of that data that you have. So one line again, just a repetition. If we are talking about null hypothesis, we are saying that there is no difference in observed and expected, and if there is a little change, this change could be just by chance. And when we solve chi-square, remember, you need to remember this formula, one thing. And another thing you need to remember is the degree of freedom. So degree of freedom is the number of ways in which the expected classes are free to vary, right? So if we have expected classes, how many expected classes we have? Either we have head or a tail, or we have tall or a dwarf. So if we have only 2 classes, then we have our degree of freedom will be 2 minus 1, which means 1. So we will talk about it when we take the example. And so there are conditions where the data could vary because of a reason. You understand that it's not always that, eventually when we will go forward and discuss about linkage, then you see that the data indeed vary with the Mendelian ratios, right? So data may not vary and data may also vary in some cases, right? And so we will take this example where a corn plant heterozygous for kernel color, a capital Y with yellow Y is self-crossed, and yellow kernel are dominant over white kernel. According to Mendel's inheritance, the expected phenotypic ratios in these offsprings are going to be 3 yellow and 1 white. And a student grew 160 corn kernels and he observed yellow kernel which are 100 in number and white kernels which are 60 in number. And we are going to use chi-square to understand if our ratios are fitting with this 3:1 ratio or if they are significantly different, right? So our null hypothesis that observed ratios fit with the expected ratios, and our alternative hypothesis is that there may be a difference for real and it does not fit with 3:1 ratios. So what we do when we calculate expected counts, we already know that we have 160 kernels, and how many times we are going to get yellow? You remember 3/4. So what will be the number if we multiply 3/4 with 160, 120, and white recessive, so 1/4 into 60, 40. And these are the expected ratios, right? So if you have 160 3:1, that is what we have calculated. And what he found is 160. So what you would do, if you would apply this formula, then you can see that you can make this kind of table where you have observed and expected, observed minus expected, and then finally the formula. Then you have the chi-square value which is 13.33. Once we have a chi-square value, as I just mentioned a second before, 13.33, we need to know the degree of freedom. And as I discussed that we already have the degree of freedom which is we calculate the degree of freedom by looking at how many classes we have in which the data can vary. So there are only 2 classes, right, yellow and white. So n is equal to 2 minus 1 is 1. So when we try to understand the meaning of our chi-square data, we need to look at the data in the chi-square distribution table. So here on your screen you can see this table. A part of this table, it's a very long table, but it is there in every book and you can also find it online. I have put a reference link for one of them. So what this table shows you is the degree of freedom at one axis, and these are different chi-square value at different significance level, right? So initially very high 0.99, and you can see it keep on decreasing to 0.005. So more stringent we will be, our data will fall in this later range of like this or this. So this is how our data, either it will be 0.5, it will be 0.5 or 0.25, 0.01, 0.005. These are the classes that if our data is more than these values, then our data is highly significant. So what you would do, because your degree of freedom is 1, you will look at 1 and then you match your value to the value that is present in 0.05 category, and the value in this category is 3.841, right? So there are only 5% chances, when I am saying that there are only 5% chances that my data is just by chance varying, then my value should be lower than this. And what we see here, our chi-square value is 13.33, which is much more than 3.84, and so the highly significant chi-square means that the deviation from the data is significant. So there is if I conclude, I will say that there is a significant difference between the observed and the expected ratios, and the deviation is not by chance. So what I am going to do? I am going to reject my null hypothesis. And so with that, I will end here with an activity question for you. The activity question is shown on your screen. The question says that a maize plant heterozygous for kernel color is self-crossed. Yellow kernel are dominant over white kernels. A student collected 160 kernels and observed 118 yellow and 42 white kernels. So now you have to use the chi-square test to determine whether the observed data fits with the expected 3:1 Mendelian ratio, or you have to test this data by checking its significance at 0.05 in the chi-square table. So with that, I want to thank you for your time and attention, and I will see you in the next class. If you have any questions, you can write me or directly or write in the chat box. Thank you.